Hello everyone, and welcome to today's webinar, Expanding ERF, Equivalent Number of Reference Fluorophore, Value Assignments for Quantitative Flow Cytometry, More Colors and Smaller Particles. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This presentation is educational and thus offers continuing education credits. Please follow the process to obtain your credits by clicking on the Continuing Education Credits window. I'd like to now welcome our speaker, Paul Derose, PhD, Senior Bioanalytical Chemist at the National Institute of Standards and Technology. Paul, you may now begin your presentation.
Hi, I'm Paul Derose, and I'd like to thank Thermo Fisher for inviting me to speak. Today I'll be talking about NIST assignments of ERF fluorescence intensity values of calibration microspheres for quantitative flow cytometry. And more recently, we're expanding these assignments to more colors and smaller particles. First, I have to show this disclaimer. The top disclaimer is Thermo Fisher's, the bottom is NIST's. And I should mention that NIST was provided dye materials by Thermo Fisher Scientific to support the work presented here. So the collaboration between NIST and Thermo Fisher is a long one. We've been collaborating with the Protein and Cell Analysis Business Unit for more than 20 years, developing fluorescence intensity reference materials and microsphere calibration standards for flow cytometry. More recently, we've had a CRADA with Thermo Fisher for assigning ERF intensity and particle concentration assignments of cell-sized microparticles. We assigned more than 25 different fluorescence channels for flow cytometer calibration. And this was completed towards the end of 2020. At about that same time, we started a new CRADA with Thermo Fisher, where we are presently doing ERF intensity and particle concentration assignments of virus size or submicron, as opposed to micron size, which would be cell size microparticles or nanoparticles for flow cytometer calibration. And this is ongoing. So the first thing that we're doing as part of this new CRADA is looking at the virus check kit beads, which are 100, 200, and 500 nanometer polystyrene beads that are intended to aid in the development of viral therapies and vaccines. And keep in mind that the COVID-19 related virus has a diameter of 100 to 120 nanometers. We believe that these joint efforts will make an important contribution to combating the pandemic and fulfilling our community responsibilities as leaders in biotechnology. So to give you an introduction, a little introduction to flow cytometry, although most of you may not need it, you're basically taking typically a cell suspension, you're putting it through a narrow channel such that the cells go through single file. So you're detecting single cells or particles using light channels. You may be detecting scattering or fluorescence. And you're counting the number of cells or particles in a particular population based on an intensity gate which defines that population with a particular light characteristic. And then downstream of the detection region, at least for some instruments, you're able to then sort cells based on those different gatings. So what are you actually measuring in flow cytometry? Well, you can measure forward light scattering, which is the intensity being directly proportional to size. You can measure side scattering, which is related to shape and internal complexity, or you can measure fluorescence. We're going to be concentrating on fluorescence today, and with fluorescence, you have a fluorescent tag, which is attached to some biological moiety. So it's typically say antibodies. So you're using the fluorescence intensity to count antibodies or receptors on cells, as well as to quantify gene expression. And we do that by measuring the mean fluorescence intensity, or MFI value, of a particular population. The MFI value is directly proportional to some biological quantity, for instance, the number of antibodies bound per cell. The problem is that MFI values are different on every instrument. And this has to do with a variety of things, mainly that every detection system has a different spectral response to light. And because fluorescence is an absolute technique, it's not a relative technique, say like absorbance. So how do we overcome this? We use calibration beads. The calibration beads are assigned fluorescence intensities, which are given by the calibration bead vendors to the customer. So you go in, you're looking at different populations of calibration beads. So for any particular channel, you have a set of beads at different intensities. For instance, if we look at the center, a green emission channel, each one of those peaks corresponds to a different bead population with a different fluorescence intensity. If we integrate any one of those peaks, we get an MFI value for that bead population. If we take the group of those and we graph them, so we're measuring the MFI value on our particular instrument, we're putting it on an absolute fluorescence intensity value shown on the y-axis, which in our case, NIST is assigning ERF values, which stands for equivalent reference fluorophore units. So we can then calibrate our instrument, the instrument dependent quantity MFI to the instrument independent quantity ERF. And if we have an unknown, which is shown by the X here, then you can say that's a cell population. You can put that cell population intensity, which you're measuring as an MFI value, onto an absolute scale, the ERF value. So just a few more examples here. If we use Thermo Fisher AccuCheck beads here, we have a low intensity bead, a mid intensity bead, and a high intensity bead. And then we have a cell population shown in red. And we see that we can either take the 3 points that we get, the low, mid, and high intensity beads, and graph them as we did on the last slide, and then we can see where the cell population intensity falls. Another way of doing it, instead of using a bunch of points, is only using 1 point or one calibration bead, and this is called a nearest neighbor calibration method, where in this case we would take the bead that's closest in intensity to the cell population, for instance, the mid-intensity bead. And then by using the ratio of the ERF value to the MFI value for the mid-intensity bead, and then multiplying by the MFI value for the cell population, we can assign the ERF value for the cell population. In this case, it's 121,253 ERF units. So just to give one more example, just so you get the idea, here we're using Thermo Fisher performance beads at 3 different intensities, a low, a mid, and a high intensity. And we are quantifying the intensity of 2 cell populations, one being a CD19 positive and the other being a CD19 negative cell population. And again, we can use the 3 calibration bead points to see where the intensities of the cell populations fall on that calibration line. Or we can use the nearest neighbor approach. In this case, the positive population is closest to the mid-intensity bead. So we would use that to do an assignment, and for the negative population, that's closest to the low intensity beads, so we would use that for the nearest neighbor assignment. So you have these two different ways to do it, and so the question you might ask is, well, how do I know which one to use? And the answer that I would give is that for most cases, you want to use a line calibration with multiple points, as this will give you a more accurate result with a better defined uncertainty. But in certain circumstances, if you had, say, a cell population with an intensity that was particularly low, say on the low end of your calibration line, or particularly high on the high end or beyond of your calibration line, then it might be more accurate in some cases to use the nearest neighbor method. So why did NIST decide to do these ERF assignments, this absolute fluorescence intensity assignment scale in the first place? And this goes back to an interlaboratory study that was done through ISAC, the International Society for the Advancement of Cytometry, back in 2016. And so NIST and 4 different calibration bead vendors each did independent ERF assignments using the same beads and the same reference fluorophores that are used to do the ERF assignment. And I'll talk a bit more about the details of that later, but the gist here is that there was poor agreement between the participants. And this poor agreement, and we're not talking about, say a factor of 2, we're talking about orders of magnitude of disagreement. And so this poor agreement led the bead vendors to strongly support a bead calibration service that would be offered by NIST. So NIST offers a variety of services to our customers. We produce reference materials through the Office of Reference Materials. We also offer calibration services where NIST is thought of as the expert on a particular measurement. We decided to do these ERF value assignments through CRADAs, individual CRADAs with NIST and a particular stakeholder, say an individual bead vendor. And the reason why we decided to do it that way is that the CRADA would enable us to address the unique needs of individual stakeholders, because every stakeholder had slightly different needs. And it's also not an exercise where NIST is looked at as the expert, but it's actually a learning curve for both NIST and the stakeholder. NIST lending our measurement science expertise, and the stakeholder lending to NIST their expertise in making calibration beads, as well as applications that are needed by their customers. So who are the stakeholders? Who have we been working with over the past few years to better quantify flow cytometry? And our collaborators have included stakeholder representative organizations such as NIH, FDA, ISAC, and the International Society for Extracellular Vesicles. We've also done ERF assignments for a variety of calibration bead vendors, including BD, Beckman Coulter, Thermo Fisher, Bangs Labs, Spiritech, and Boston Cell Standards. And we've also worked with other stakeholders, including those in clinical testing labs, such as at Tufts Medical Center and the Canadian Immunohistochemistry QC Laboratory. We've also worked with more than 16 biopharmaceutical companies who are interested in monitoring their online water, which they use to make their biopharmaceuticals. And the idea is that they don't want to have microbes in that water, because it obviously could affect the effectiveness and the shelf life of their pharmaceuticals. We've also worked with other collaborators for other types of quantitative applications, including BioLegend, Fluidum, and IBSC and LGC. We've also been involved in the development of documentary standards, such as the CLSI standard guidance document H62 for validation of assays performed by flow cytometry, as well as the USP chapter 127 for flow cytometric numeration of CD34 positive cells. So how exactly does NIST do an ERF fluorescence intensity assignment of calibration beads? Well, it's a two-step process. The bead vendor sends us their calibration beads in a bead suspension, and we take that bead suspension and do two measurements on it. The first measurement shown at the top is a fluorescence intensity measurement, where we relate the fluorescence intensity of the bead to the fluorescence intensity of a reference fluorophore solution. And then shown at the bottom, we also do a measurement of the number concentration of the bead suspension. So we put these two values together and we get an ERF intensity assignment per bead. The three references shown at the right in blue go into more detail on exactly how we've done this, and the most recent one, which came out the end of last year, appeared in the journal Materials, is probably the most current and the most detailed. So what these ERF assignments give us are fluorescence intensity values for a particular flow cytometer fluorescence channel under a specified set of instrument conditions. And these ERF values are comparable between instruments and over time, even when different manufacturers' calibration beads are used. So we're basically going from beads that are instrument dependent before their assigned ERF values to values that are instrument independent. So first, we're going to take a little closer look at the fluorescence intensity measurement. We do this on a fluorescence spectrometer, not a flow cytometer. And what we measure is the fluorescence intensity of a reference solution. There are 4 reference solutions shown here, which are part of SRM 1934. And we compare the fluorescence intensity of one of these reference solutions to the bead suspension calibration bead intensity. So for instance, if 10 to the 5th molecules of a reference fluorophore fluoresce with the same fluorescence intensity as one calibration bead, then we can say that that calibration bead has an ERF intensity value of 10 to the 5th ERF units. So we're using a NIST home-built fluorescence spectrometer that's calibrated for spectral shape and linearity, which gives us corrected spectra for both the reference solution and the calibration bead. So there's an example shown in the graph here. We have Nile red as a reference fluorophore, and we have a Percy P labeled bead that we're assigning. So we measure the two spectra, and we then integrate the spectra over the bandpass filter spectral region that would be used on your particular flow cytometer. And that gives us two intensities that are equivalent to the intensity that you would measure on a flow cytometer. So we compare those two values. We have a serial dilution of the reference fluorophore solution, and shown with the blue points here. The blue points give us a calibration line of fluorescence intensity in terms of reference fluorophore concentration. So it's important to note here that ERF actually expresses fluorescence intensity in terms of reference fluorophore concentration. So it's not actually what you would typically think of as a fluorescence intensity. And we then take the bead suspension intensity, put that on our calibration line, and are able to deduce the fluorescence intensity of the bead in terms of the reference fluorophore concentration. So we have 4 dyes which make up SRM 1934, coumarin 30, which covers blue emission, fluorescein, which covers green emission, Nile red, orange emission, and APC red emission. And one dye covers a particular spectral region with multiple fluorescence channels. So you don't need a fluorophore, a reference fluorophore for every channel, or for every fluorophore that you would need on a bead. And this is an important point which differs it from MESF, for instance. We also measure the bead number concentrations. The primary method that we use is light obscuration, and at least for micron-sized particles. And we decided to use light obscuration because it's a simple technique. You're basically looking at a dip in the laser intensity as a particle passes through the laser. And what's great about this technique is because it's so simple, we can easily get at the uncertainties that are involved in the technique, and therefore can specify what those uncertainties are. We've shown this technique to be an SI traceable particle counting technique, which has been shown in detail in the reference shown at the bottom in blue, which appeared in 2018. We also use other techniques to support these measurements, kind of as a reality check. We always use flow cytometry to back up the light obscuration measurements. We used true count beads as an internal standard in that case, and we've also used a Coulter counter to compare measurements. And when we do compare the measurements, we find the values to agree within about 5%. So what are the final values that we give to our customers? We give ERF values with total uncertainties reported. The ERF values are, in most cases, are SI traceable, and the total uncertainty that we give is a 2 sigma 95% confidence level uncertainty, which has ranged from about 5% to 13%. We also give each customer a NIST certificate. This is an official NIST document which tells the ERF value for a particular bead and fluorescence channel and the related uncertainty. So one thing I wanted to emphasize here is that once we have ERF values for calibration beads, we've put those fluorescence intensities onto an absolute scale, but it's an absolute fluorescence intensity scale. It is not an absolute biological scale. So still, to put the ERF value scale, to put the ERF scale onto an absolute biological scale, say antibodies bound per cell, we still need at least one biological standard, which is shown here. We're using one biological standard to put ERF shown on the left axis to ABC on the right axis. We've expanded the number of reference fluorophores that we use for assignments beyond SRM 1934. 3 new ones include Pacific Orange, which uses the same excitation wavelengths as coumarin 30, but goes further into the red side. It goes into yellow-green emission, as well as Alexafluor 700 and Alexafluor 750, which go further into the near IR than APC. So you remember that I said that we use these reference fluorophores to express fluorescence intensity in terms of reference fluorophore concentration. In order to do that, we have to determine the purity of the fluorophores. So I just wanted to give an example of that with the new dyes. So here, for instance, is Pacific Orange, and we initially use HPLC kind of as a quick and dirty to show that it's approximately as pure as we need it to be a reference solution and a reference fluorophore. So using HPLC, we found the Pacific Orange to have a purity of about 1%, an impurity of about 1%, so a purity of about 99%. We then use quantitative proton NMR as an SI traceable purity technique. So this is really our primary SI traceable technique for determining purity. We use a NIST primary standard for quantitative NMR benzoic acid, and Some of the issues that we found with the 3 new dyes that we didn't find with the original 4 dyes in SRM 1934 is that the new dyes were very hygroscopic, so we had to do gravimetry under dry nitrogen, which complicated things a bit. We also found that the counter ion and dye molecule ratio was not an integer for these dyes. And because of that, we had to determine purity for the 3 new dyes in terms of grams of dye molecule per gram of solid sample, as opposed to say moles of dye per sample, which we were able to do for the original SRM 1934. We also found that the impurity values that we found for quantitative NMR were significantly higher than what the HPLC implied. And we saw as much as 6% for Pacific Orange, 4% for Lexaflor 700, and 13% for Lexaflor 750. And because of these discrepancies between the HPLC and the quantitative NMR, we do have high confidence as to the accuracy of the quantitative proton NMR technique in this case. But since we don't know what is causing this discrepancy, what are the unknowns that we haven't identified, the purity values become reference values instead of certified values, certified value being a higher level of confidence according to NIST criteria. So here are all the dyes. Here's our palette of colors of reference fluorophores that we've used to this point. The larger peaks being the fluorescence emission spectrum of each dye, which defines the fluorophore channels, the fluorescence channels that we can cover using a particular reference fluorophore. And then the smaller peaks are the absorbance spectra for each of the dyes, which defines which laser wavelength or wavelengths would be used to excite that particular reference dye. What we want to do in the future is really cover all of the fluorescence channels that are available on flow cytometers. So we want to go further into the UV on the left side and further into the near IR on the right side, as well as maybe filling in some gaps in the middle. And we are working on that. And this is just a tabular format of what you just saw in the graph. So I wanted to give you an idea of the different fluorescence channels that we've covered to this point. So I listed all of them. And using coumarin 30 as a reference fluor, we've assigned 10 different fluorescence channels. Using Pacific Orange, we've assigned 6 different fluorescence channels. Using fluorescein, we've assigned 8 channels. Using Nile Red at 488 excitation, we've assigned 7 channels. Using Nile Red at 561 nanometer excitation, we've assigned 10 channels. Using APC, we've assigned 6. And using Lexaflor 700, we've assigned 4 channels. So in summary, of all the ERF assignments that we've done to this point, we've assigned more than 50 fluorescence channels using 5 lasers. 6 are now available to us, with emission ranging from 390 nanometers to 850 nanometers. Using 6 reference fluorophores, 7 are now available to us. And we've covered diameters of calibration beads from 2 microns to 10 microns. So where are we going next? We're going to smaller sizes. We want to characterize submicron or nanometer-sized particles in a similar way. This is of great interest because of the study of extracellular vesicles and viruses, which have diameters that typically range from 40 nanometers to 1,000 nanometers. EVs in particular are thought to mediate intercellular communications in physiology and pathology. And many of the same surface receptors as cells are found on EVs. So because of that, this is a topic of growing interest in flow cytometry. Now, in principle, the same approach that was used to do ERF assignments for micron-sized particles can also be used for nanometer-sized particles. The problem is that there aren't techniques that have been well established to be accurate measures of bead number concentration. So it's not the fluorescence intensity measurement that we see as the problem here, it's the bead number concentration. And the few examples that you find in the literature, such as electron microscopy, tend to be very time-consuming and very difficult and very expensive. So we need some easier, more user-friendly techniques. And before I go on to speak about that a little bit more, I just wanted to acknowledge the people who have participated and contributed to these ERF assignments at NIST. This includes Lili Wong, Dolph Gagulis, Linghua Tian, Dean Ripple, Sandra De Silva, and Nancy Lin. We also, Dolph Gagulis has done modeling of antibody binding, and the Quantitative Proton Nmr was done by Aaron Urbis and Mike Nelson. And we also have groups at NIST which meet periodically where we have particle counting discussions and also submicron bead number concentration measurement discussions. And this latter group is also doing all of the measurements that I'll be talking about next. So as I said at the beginning, the first nanometer-sized particles that we'll be looking at is part of the virus check kit from Thermo Fisher. And this kit is meant to aid in looking at and the development of viral therapeutic and vaccine investigations by helping to improve flow cytometry assay accuracy and efficiency, ensure intra and inter-laboratory data comparison, and to improve day-to-day reproducibility. The proposed virus check kit is comprised of 5 different components. Each of them is a 0.5 milliliter bead suspension with a concentration of 5 × 10⁶ beads per mil. The 5 components are 3 100 nanometer beads labeled with FTSE-like, PE-like, and APC-like fluorophores. And then we have a 200 nanometer bead and a 500 nanometer bead that are labeled with multiple dyes, so-called tetraspec beads. So what are the different techniques that we're looking at NIST? Well, we're looking at electron microscopy, particularly SEM. Now, I did say this is probably the most difficult and time-consuming, but to this point, it's kind of been looked at as the gold standard. So we thought we should definitely include it as a comparison. It gives accurate core sizes and relative number concentrations, but doing absolute concentration measurements is very difficult. We're looking at asymmetrical flow, field flow fractionation with dynamic light scattering detection. AF4 is used to separate particles by size. The AF4 allows the dynamic light scattering measurement to be more accurate. And some of the problems that you have with AF4 with DLS is that relative number populations are biased toward larger particles. DLS is also not quantitative for absolute number concentration. But AF4 can be combined with other detection techniques such as UV absorbance and multi-angle scattering to make number concentration more quantitative. So we're working on that. We're also looking at nanoparticle tracking analysis, or NTA. It's an optical method which takes snapshots of moving particles in solution. It's an optical method which measures the hydrodynamic radius of particles. One of the problems is that you need a number concentration reference standard for an accurate absolute measurement, and large particles mask the detection efficiency of smaller particles in mixtures. We're also looking at resistive pulse sensing, or RPS, also known as a Coulter counter. which gives accurate size diameters and relative number concentration, but again, it needs a number concentration reference standard to do absolute measurements. And the size limit here is about 200 nanometers, so you can't go below 200 nanometers. There's so-called next generation RPS, which can use microfluidics and other techniques to get to smaller microliter sample sizes, and also to get to smaller diameters as low as 60 nanometers. Again, the same problem arises. You need a number concentration reference standard to do absolute measurements. We're also looking at nano and quantum flow cytometers. We have one that was built at NIST, and the size, it promises size limits of 30 nanometers or smaller, but these are not commercially available, and we don't have enough data to say much at this point about accuracy. There are also virus counters. The one that we have at NIST is basically a specialized flow cytometer with 2 fluorescence channels. It's designed for virus detection. It's meant to handle size range from about 25 nanometers to 300 nanometers. And one of its advantages is it measures volume in real time. We don't have enough data to say how well it will do with detecting beads, and also how accurate is the real-time volume measurement. And then the last technique that we're looking at to this point is a type of fluorescence microscopy where we use charged slides so that the beads or particles stick to the slides, and combined with a more accurate way to do sample volume determinations. And this is similar to the way you would use a hemocytometer, but it gives greater accuracy, precision, and speed. And the size limit here is about 80 nanometers. We have one at NIST, but it's not commercially available. And then here's basically a summary of everything I just said about the different techniques, going over each technique, specifying the size limit and size range, the optimum sample volume for the particular technique, the optimum concentration range for the technique, and caveats. So far, we've looked at numbers from 5 of these techniques. We've compared them, looking at 100 nanometer and 200 nanometer beads. Right now, we're seeing agreement within a factor of 2, but that's not good enough. I'd say we'd like to go, say, under 30% uncertainties. So we're working to eliminate bias in the different techniques and understand the uncertainties involved in each of them. So the important developments here is that flow cytometry quantitation, the consortium, and the CRADAs that we've developed bring stakeholders together to develop measurement services, reference materials, reference data, and reference methods. And this is a NIST and NIH co-managed consortium. I should also mention that the Flow Cytometry Quantitation Consortium has recently just been replaced by the Flow Cytometry Standards Consortium, where we're expanding the scope of the consortium beyond ERF measurements. NIST is performing ERF assignments of calibration beads through CRADAs. We're adding more reference dyes and laser colors to assign ERF values for all fluorescence channels that are available on all flow cytometers. That's our ultimate goal, with the ERF at least. We're also working with stakeholders to determine number concentrations of submicron bioparticles and assign ERF values to submicron calibration beads. So one of the ways that we're trying to expand our scope is to consider antibody reagent quality. And there was a report that came out from a working group that appeared in Nature Methods in September of 2016, which reported that that there at the time were 300 companies that sell antibodies. There's probably more now. And antibody reagent quality was found to be inconsistent and sometimes poor. And many clinical diagnostics and therapies rely on such antibody reagents. So it was estimated that more than $800 million per year is wasted due to poor quality antibodies. And what was suggested to going forward is that what was needed are reagent and documentary standards to help determine quality and ensure reproducibility. So part of our expansion of scope includes opening up fluorescence and other types of measurements in order to better characterize antibody reagents, including looking at fluorescence quantum yields, fluorescence lifetimes, and corrected fluorescence spectra. We're doing thermodynamic modeling of antibody binding characteristics. We also would like to do a flow cytometry standardization round robin where we look at ERF assignments of reference cells. Just some of the things that we have in the pipeline. And with that, I thank you for your attention and would be happy to take any of your questions.
Thank you, Paul, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, to estimate antibodies bound per cell, I believe it is important to have a 1-to-1 fluorophore to protein ratio. When you use other fluorochromes, how does this compute?
I would say that, you know, if you know, right, the number of fluorophores per antibody, that helps to get you to some biological quantity, but it's not necessary just to do a calibration of your instrument. So it depends on what you're trying to achieve with your calibration beads. I guess you can certainly, I mean, that's part of why we're doing modeling, antibody modeling, so that we have a better understanding of how exactly is fluorescence intensity related with the number of antibodies that are there. And even for simple cases, it's not totally straightforward to assign a biological quantity using a bead, because, you know, the bead chemistry and the biological, say the cell chemistry, can be quite different. You know, also some beads are hard dyed, some beads are surface labeled. You know, hard dyed beads probably aren't particularly useful to get biological quantities, but surface labeled beads may be more useful in that way if you have an understanding of things like you said, the number of fluors per antibody.
Thank you, Paul. Our next question. Can you explain again how this differs from MESF beads?
Yes. Right. So MESF, which stood for molecules of equivalent soluble fluorophore or fluorochrome, as opposed to ERF, which is equivalent reference fluorophores. The difference is that MESF is a special case or subset of ERF. So MESF means that the reference fluorophore and the bead fluorophore are the same. So the two fluorophores for ERF, in contrast, cover the same spectral range, but don't have to be the same dye. So ERF is a more practical choice because you don't need a different fluorophore for every existing fluorescent label. You know, there are hundreds of fluorescent labels, and so it's impractical, and it's certainly not within NIST's budget to do that, even if it was possible, even if it was chemically possible, which in some cases it's not.
Thank you, Paul. Our next question here is, you presented the interlaboratory performance study done in 2018. Can you discuss studies done since or planned that will address the value of ERF assignments to improve the FI reproducibility issues? Or FL, probably fluorochrome fluoride.
Fluorochrome reproducibility issues. I mean, we are doing additional studies. I'm not sure what the question is referring to as far as fluorescence intensity reproducibility issues. I guess they're talking about the study. So right. So they're saying that are we trying to make materials available so that people can actually do their own ERF assignments? I mean, that's what we tried to do in this original study, and that's why NIST is doing the assignments, because there was too much disagreement, and we also did identify some systematic errors in the approaches of other laboratories, but yeah, I mean, I don't see at least not in the near term, a lot of other people doing ERF assignments besides NIST, even though it certainly is possible, and we encourage people to attempt it. But I think what we are doing is we're encouraging. We want to do a round robin where we take calibration beads, we pass them around, and make sure that different calibration beads can give the same answer on different instruments. And because that to me is the most important thing that we have to show. It's the whole purpose of the ERF assignments. So until everybody can do that easily, I think that's the priority.
Thank you, Paul. And I believe the questioner has just clarified his question. Have other interlab studies been done with ERF to improve?
We are working with certain laboratories who are interested in doing their own ERF assignments, but so far we've been doing that on a one-on-one basis because at least to this point there didn't seem like a large interest from the original participants to quickly do this kind of round robin again.
Okay, thank you, Paul. And our next question here. Can I use Accucheck ERF particles as intensity reference for a spectral flow cytometer? I have a Cytec Aurora.
Yes, the answer is yes. As long as the spectral range used to determine the fluorescence intensity is the same for the spectral flow cytometer and the calibration bead. So you want to make sure that if you have a spectrum, then you have to integrate over a particular gate that you choose to define a fluorescence intensity, and that gate just has to be consistent between the instrument and the bead.
Thank you, Paul. I have time for a few more questions. Can I use ERF particles to compare exosome data?
The answer is yes. That's why NIST wants to assign ERF values to smaller beads. If you use cell size or micron size beads for your exosome assignments, you're going to get inaccuracy. I don't even think that level of inaccuracy is completely understood or has been has been shown exactly what it is, but certainly even theoretically we don't expect it to be as accurate as it would be if you had micron-sized beads to do the calibration.
All right, and we've got one more question. Do ERF beads replace MESF beads?
I mean, I guess that question, I guess, is similar to, I guess, what it was the first question. I mean, I guess I already explained the difference between MESF and ERF. I mean, the simple answer to that question is yes. You know, I think, you know, for all the reasons I stated, just because of the practicality of using ERF versus trying to have a bead for every, a different reference fluorophore for every type of fluorescent label that you would find on a bead or a cell.
Thank you, Paul. Do you have any final comments for our audience today?
I think you know, as you saw from our from the acknowledgments, you know, there's a lot of people at NIST working on this. We also have a lot of collaborations with people outside of NIST, particularly looking at the submicron regime, and you know, so we're excited about this work, and we expect to have some good results in the near term. You know, I would also you know, like to thank again everyone who attended the talk today. And if you have any further questions that pop up, you're welcome to email me. My email is on the NIST website, and it's also just first name dot last name at nist.gov. So thanks a lot.
Thank you again, Paul, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.